Good morning, everyone. This is section 3 of chapter 4, eukaryotic cells. So, eukaryotic cells are plant and animal cells. A lot of the times, people get confused with prokaryotic versus eukaryotic and plant versus animal. Okay, prokaryotic is a whole different thing. Eukaryotic encompasses animal and plant. All eukaryotic cells have a plasma membrane. We already kind of talked about that. A nucleus and membrane-bound organelles. So all these more developed organelles are in these kind of cells. Okay. So one of the things your books tells you is that cells are systems. They are like little loyal soldiers to you in your body. So they will even kill themselves off if they think that's going to benefit you or the organism that they're living in. Below is a plant cell, and on to the right is an animal cell. We're going to go over all of the organelles in a eukaryotic cell. So organelles are just these guys that live within a cell, and they're membrane bound most of the time. Okay, so the first one is vesicles. So I always try to come up with something that reminds me of that organelle, so because my mind is going to process that information a little bit better. So vesicles are membrane sacs that enclose molecules and keep them separate from the cytoplasm. So these are the taxes of the cell, okay? So everybody look up at the board. You can see that these little guys, these little green looking little, they look like little bitty fish, but they're not. These little green particles on the outside of the cell are wanting to come in. So they're pushing on the cell. All they do is push on the membrane. You can see, you can see the phospholipid bilayer, and it's just going to push until eventually it's going to pinch off like Play-Doh and become a vesicle. Okay, it's membrane bound. It's separating what's inside of it from the outside. Next up is the cytoskeleton. The cytoskeleton is an extensive network of protein fibers. These things are like the highways of the cell. It almost like directs traffic and directs organelles, vesicles, proteins, anything else that are moving about the cell into their correct place. It's also going to help with cell shape. Okay, that's where the skeleton term in the name comes from. Cyto, because it's in the cytoplasm. Sorry, I know sometimes I go too fast. Okay, so again, remember, plant cells are eukaryotic cells too. Why? Because they have a nucleus and because they have membrane-bound organelles, okay? They have three things that separate them from animal cells. I will ask you this. They have a cell wall, a chloroplast, and a vacuole. If you want to put central vacuole, that's fine, and you can think of the 3 C's. Because there are vacuoles out there, but they actually have a big one, and it's that big bubble looking thing right there in the middle. It's almost like a water tank. If you want to think about it, if you want to put central at the beginning of that, that's fine, and you can think of it as the 3 C's: cell wall, chloroplast, and central vacuole. Those are the 3 things that separate a plant cell from an animal cell. You're thinking, what is a chloroplast? That's fine. We'll go over that in a minute. Okay, so section 4, nucleus and ribosomes. So we're going to talk about how proteins are made. The nucleus is the command center or the brain of the cell. It's located usually near the center of the cell. So while you're writing this down, I want you to think in your brain, in your mind, just think to yourself, what does the nucleus do? What do you think the nucleus does? So the answer is very simple. It just houses the DNA. It's almost like the protector, like your skull almost. It just protects the brain, protects the DNA. There are parts to the nucleus that we're going to go over. So don't think about this as the nucleus. This whole purple thing is the nucleus, okay? Don't look at it as a cell. This whole thing right here is the nucleus. So first you're going to have the envelope, which is the double membrane, okay? It's just this purple thing on the outside. The nuclear envelope is the double membrane. You also have nuclear pores. These are little, just like pores on your nose. This allows passages, or allows passage of ribosomal subunits. These are passageways almost, so that small things can get in and out. The particular things can get in and out. mRNA is one of those things. So mRNA is really small. It goes in through the pores and it reads a piece of that DNA, whatever instructions are needed, and then it goes back out of the pore and takes it to the ribosome. It's really small. It just gets a snippet of that cookbook almost, and then takes it and makes that recipe. These pores act as gatekeepers to regulate what comes in and what goes out. All right. Let's look at them. So you can see these little dots are the nuclear pores. Then you have the nucleolus, which is even denser. It's real deep within the nucleus. And they actually make ribosomes, and the ribosomes get sent out too. I'll give you just a second to write that. Okay. It's another picture of a nucleus. You can see the nuclear pores on it. And it imprisons the DNA, doesn't allow it to get out. The nucleus also has chromatin. So chromatin is uncondensed DNA, okay? So this is just relaxed, like you're sitting home on your recliner, on the couch, in your pajamas, okay? You're not wound up. But when the time comes, right, you have a job to do, and you might have to put on your work clothes, button those pants, and get to work. So that's what chromosomes are. Those are condensed versions of genetic info. And that only happens, it only condenses when it's about to do its job and replicate and pull apart, okay? That's where genes are located. So just like I just explained, chromatin coils up into these rods, into this chromosome, just before cell division. Then when it's done, it goes home and it relaxes and it uncondenses back into chromatin. It's just two different names for the same thing. So you can see right here, if we break it down, we learn about this type of DNA right here, which is the same DNA all the way through, but it's just coiled up and wrapped up tighter as you go. So these chromatin, they're going to have maybe some histones, or something that the DNA is wrapped around, but they're still relaxed. But when it comes time to do their job and replicate and pull apart, they're going to condense down into this X structure chromosome. We're still talking about the nucleus, okay? I think we're almost done with this section. So there are 3 types of RNA produced in the nucleus. Remember that nucleoid region makes ribosomes. The nucleus is going to also make RNA. You have 3 types. You have rRNA, which stands for ribosomal RNA. It's going to be involved in protein synthesis. You have mRNA, which stands for messenger RNA. This type of RNA reads DNA and takes a message, like I talked about going through that pore, to make the protein, whatever protein that DNA tells it to. And then the third kind is tRNA, and it stands for transfer RNA. And this is going to aid in protein synthesis. It's going to take amino acids and puts them together in the correct order to make your polypeptide. So this whole thing is chapter 12. So you need to make sure that you are paying attention and focused now, because we're just going to dive deeper into parts of this. Okay. So on the top left, everybody look at the board. On the top left, you can see that mRNA has broken up that DNA strand, and it's reading parts of the DNA. It's only going to be really short, the mRNA is, and it's going to take that message that it just read and go to a ribosome, so transfer her like it's over here. So you can see the mRNA on the bottom right. That's it right there. Okay, there's its little bases that it read. This ribosome is made up of rRNA. And then you have these tRNA, these little weird T-like structures that have an anticodon on them, and they have the amino acids, and they actually, based off of what the mRNA says, we'll go over all of this later. The tRNA puts the correct amino acids together, which makes the peptide. All right, so one more portion of this, then we're going to do section 5 and be finished for the day. So ribosomes. So ribosomes are particles. They're not membrane-bound organelles, okay? They're particles where protein synthesis occurs. You can find ribosomes in eukaryotic cells and prokaryotic cells. The number of ribosomes that you might have in a cell vary. They're located just scattered about the cytoplasm, and they're also attached to the endoplasmic reticulum, or we call it the ER, so we don't have to type that or write that every time. So that's what makes it rough ER, okay? That's why they call it rough ER and there's smooth ER. The rough ER is going to have ribosomes on it. Okay, you can see, if everybody look at the board, it's the point of putting pictures up here, okay? You can see these free ribosomes right here, these tiny little black dots just kind of floating, but most of them are on the rough ER, and that's what actually makes it, in quote, rough. Over here in orange, that's the smooth, that's the Golgi. The smooth ER, where's it at? Oh, right here. The smooth ER is over here on the bottom. It's purple in this picture as well. It doesn't have any ribosomes on it, so it's smooth, and it has a whole different function. You can see the nucleus. You can see the nuclear pores. You can see the nucleolus deep down in the middle. And so far, that's everything that we have went over. All right, we're going to talk about one more section in this part of the notes, and then we'll be finished. So we're going to talk about the smooth and rough ER. Okay, we're going to talk about the endomembrane system in section 5. So endoplasmic reticulum, or for short ER, is a membranous channel and sac that's going to be really close to the nucleus. Okay, parts of it are going to be continuous with the nuclear envelope. So if you look really close at the picture, you can see where the nucleus and the rough ER are going to be attached. This is because that mRNA goes and reads that DNA in the nucleus, and it can just go straight really quickly to the ribosome. It's just more efficient. And like I've said before, there is rough ER and there's smooth ER. All right, so the rough ER, it's studded with ribosomes on the side that faces the cytoplasm. It's only going to be on one side. Proteins are made here because there's ribosomes on it. So take note that ribosomes make proteins, okay? But the site where they are made is the rough ER. So think about school. The school building is where you learn, right? But the school building itself did not teach you anything. The teachers hopefully are teaching you something along with yourself and your responsibility. So that's what the rough ER is. It's almost just like the school that houses the learning, or the school that houses the protein synthesis. So inside these little folds, inside the lumen, these proteins are they've already been made, okay, but they're folded and they're like packaged and getting ready to go. Almost like fine-tuning the proteins. The smooth ER is also continuous for the nuclear envelope in the rough ER, but it does not have ribosomes on it, so it's smooth, okay? Some cells have more than others, just depending on what organ they might be attached to, or organ they might be, what organ they make up. So smooth ER has many different functions. It can help produce lipids, or it can detox from drugs and alcohol. So what organ do you think has a ton of smooth ER? I hope you said liver. So this is how sometimes people that are alcoholics or are addicted to drugs, sometimes this is how they overdose. So what happens is they're just pretend like they're on drugs, and their smooth ER over time has developed their liver over time, their liver cells have developed a lot of smooth ER, right? Because they're doing a lot of drugs. The smooth ER is keeping up with it. They're detoxing them, right? They're detoxing. Well, then they go to rehab. So your cells are really efficient. So those liver cells are like, oh, she's better, right? There's no point. This smooth ER is just sitting here now. So the smooth ER will almost disintegrate, right? It'll just go away. So your liver cells don't have a lot of smooth ER anymore. Well, then that that girl or whoever it is in rehab. Well, she goes and she relapses, and she does just the same amount of drugs as she was used to, because that's what she thought, was going to get her that high feeling. And what happens is your body doesn't have that smooth ER anymore, so you actually poison yourself, and sometimes you end up passing away. It's because the smooth ER in your liver cells. Same thing with alcohol. And that's why over time, like maybe when somebody first starts drinking, if they drink, they only drink a little bit and they feel drunk. But after a year of drinking, that amount to get that same feeling has grown. Why? Because the body is making a lot of smooth ER to try to keep up with your alcoholic intake. So somebody that's been drinking for a long time has to drink a lot more. Those smooth ER organelles are just working themselves to death. So people's liver looks a little different than others. So the endoplasmic reticulum is smooth and rough. Both form vesicles to transport molecules mainly to the Golgi apparatus. Sometimes this is called the Golgi body. So this picture, you see the nucleus in purple. You see the rough ER in pink. And you see the smooth ER in orange. Okay, they're all connected. All the membranes are connected. You can see the ribosomes are studded on the pink. So the last thing, almost, in section 5 is the Golgi apparatus, or the Golgi body. These are curved, flattened sacs. They're not attached, okay? It almost looks like a stack of pancakes. You're going to see a lot of vesicles around the Golgi. So the Golgi is going to alter substances or proteins as they move through the sacs. It's going to sort them, pack them, and send them off. So in my brain, I think about the Golgi apparatus as being like Amazon or UPS or something like that, or USPS or whatever you want to think of, our mail. Because think about Amazon. So if I get on Amazon and I want a pair of new Nike shoes, I'm going to order them from Amazon because I'm lazy and it's easy. I click it and I order these Nike shoes for myself, and they're coming my way in 2 days. Did Amazon make those Nike shoes? No. Did Amazon maybe go get them off the shelf, pack them up, put them in a box for me, and send them to me? Yes, that's the Golgi apparatus's job. Just like our world. It's nuts to me how similar our world is compared to how cells function. So the Golgi apparatus is my Amazon, or my postal service. So you can see the nucleus. You can see the rough ER. You see these proteins right here. This is a transport vesicle. It's going to take it to this Golgi. Okay, you can see the proteins are these tiny little purple dots. They're going to be sorted, packed, folded, whatever, in these little pancakes. And at the end, just like on the membrane earlier, the vesicle is just going to pinch off like Play-Doh and take it wherever it needs to go. It's going to go outside the cell here. I think this is the last one. Yes. So the last thing we're going to talk about today is lysosomes. So lysosomes, in my mind, are the grim reapers of the cell. They're just membrane-bound vesicles that contain an enzyme that breaks down useless material. So we talked about the smooth ER and somebody that might drink a lot of alcohol. When they stop drinking, that smooth ER is not needed anymore. So the lysosome comes over and kills all that smooth ER. He just breaks it down. So essentially, they, in quote, kill things in the cell. But the participants are willing. because cells are there for the organism that they're living for. So, of course, white blood cells have a lot of lysosomes because they digest foreign bodies. So Tay-Sachs disease is where you're missing a lysosome enzyme. So their nerve cells eventually die off, and they usually they only live about 3 to 4 years old because they do not have the enzyme in their lysosomes. They essentially don't have lysosomes. So this is really, really important to live. All this is essential to life. If you think about it, like you're just a big bunch of cells that are moving around. It's just weird to me. Like my cells are actually telling me to talk to you right now. It's just nuts. So again, this is just another representation of what is called the central dogma. The nucleus, then you're going to take the message, go to the rough ER, make the protein. The proteins are these little purple dots going to be transported in a vesicle to the Golgi. The Golgi is going to sort them, pack them up, put them in a package for me, and they're going to exit out, okay, of the plasma membrane. Sometimes, if we have some rotten apples of the bunch, they're going to be deferred to the lysosomes, and the lysosomes are going to eat them up and get rid of them because they were bad. And that's it.